Hear me now?
Yes. Okay.
Perfect. Okay. Thanks, Adrian, for giving me the opportunity to present some of the work in our lab. And thanks, Jody, for the nice talk. This has been a really great session while we're all working from home during COVID-19. And so my lab is broadly interested in trying to understand how cell cycle state affects cell behavior, and beyond just the worm that we all love, the elegans, I'm sort of broadly interested in how cell cycle state, particularly during interphase, affects cell behavior across the animal tree. And so I sort of became interested in this from sort of digging into literature from our own work and lots of other interesting work, where we find that, of course, most cells spend most of their time in interphase, in either one of two gap phases or when they replicate their DNA in S phase. And some cell behavior seems to be tied to some cell cycle states. And so I'll tell you a little bit about the end about one of the main projects in our lab, where we look at the invasive capacity of the C. elegans anchor cells. It invades into the vulval epithelium. And we've shown work from my postdoc and work in my own lab has shown that this is a G1 or G0 regulated behavior. The neural crest in vertebrates delaminates from the neural tube during early vertebrate development. And it's actually quite sort of controversial at what cell cycle stage this happens. From some live cell imaging, it suggests that this may be happening at the onset of S phase. In the posterior tail growth in zebrafish, the neural mesodermal progenitors actually undergo an EMT at G2. This is different from an EMT that seems to sometimes happen in G1. And then lastly, behaviors like convergent extension, where cells have to crawl together to elongate the paraxial mesoderm, as this nice DIC movie from Ray Keller's lab is showing. These cells are actually arrested in G2 when they're doing this. And so hopefully you can appreciate that there seems to be a variety of cell behaviors and a variety of cell cycle states that may be tied to these cell behaviors. And that's something that my lab is really interested in exploring. And then of course, as cells exit the cell cycle, there are sort of different flavors of G0, from quiescence, differentiation, and senescence. And so I'll give my first sort of acknowledgement slide up front, and that is that this project's been sort of one of the first projects that started my lab. So it's been going on for about 6 years now. And it was spearheaded by my first PhD student, Abraham Corman, who graduated last week, and Becca Adike, a postdoc in the lab. And then of course, there's been contributions from basically everyone else in the lab over time. And so if you want to visualize cell cycle state live, you need some sort of sensor to see that, besides just imaging histone or just by DIC. And so one of the first cell cycle biosensors that was made was FUCCI, which stands for fluorescent-based ubiquination cell cycle indicator. And this movie from the original Cell paper shows dividing helix cells labeled with the FUCCI transgene. And so these cells are reading out the activity of two complementary DNA licensing factors, CDT1, which is red in this movie during G1, G0, and geminin, which is green during S phase and G2. So in this movie, red means stop and green means go. This paper was published in 2008, and so not surprisingly, many researchers put these constructs, or varieties of these constructs, into their favorite model organisms. And so here is shown the development of the FUCCI fish. As you can see, as the somites develop, you can see the clearly demarcate the somites in red, and the example of retina in red as well, and the zebrafish eye. And so the FUCCI system has been great, but it does have some limitations, particularly if you're interested in trying to correlate specific cell cycle states with cell behavior, as we are. And so, for example, it's hard to distinguish S from G2, as the biosensor under geminin will be expressed in both phases. And then you can't distinguish a cell that's cycling in G1 from a cell that's quiescent or differentiated in G0. And so fortunately, a biosensor was developed by Tobias Meyer's lab, and Sabrina Spencer, who's now a PI at UC Boulder, published a landmark Cell paper in 2013 showing the activity of a ratiometric biosensor that reads out CDK2 activity in mammalian cell culture. And so the way this biosensor works is that it contains a fragment of a DNA helicase B, which I'll refer to as DHB throughout. It's a mammalian helicase that's the way this biosensor works is that it contains 4 serine phosphorylation sites. And so as CDK activity increases and phosphorylates this biosensor, you occlude this nuclear localization signal and expose this weaker nuclear export signal. So as cell cycle progresses and activity of the CDK increases, you get an increasing amount of the biosensor in the cytoplasm. And so the way we design is that we include a viral peptide to also co-express a nuclear mask under a histone, so we would always know where the nucleus is for quantification. And so the nice thing about this biosensor then is that as it's ratiometric, you can quantify in Fiji or image J the amount of signal in the cytoplasm, the amount of signal in the nucleus, and then put a number to cell cycle state. And so this is one of Sabrina's movies from her original paper. And so here we can follow this cell in blue, which is going to start out with high CDK activity, so in G2, because you see that you see the signal mostly in the cytoplasm. That cell is going to quickly divide right now. And then that cell is going to immediately go into the cell cycle again. So you can see it going to S, back to G2, then it's going to divide again. And so what was really fantastic about this paper is that Sabrina was able to show in an asynchronous population of MCF10A cells in the cell culture that some cells exhibited different patterns. They divided and they exited into a CDK low state where they had high nuclear signal, and they hung out in that state a while, and then they eventually went on to divide. And so in her cell culture system of randomly growing cells, she could distinguish two populations of cells, and you could distinguish this by the activity of that biosensor. And so if you take all of those traces and plot them, you see that she has this bimodal distribution of cells that are born into a CDK2 increasing state that go on to divide sooner, and cells that are born into a CDK2 low state that go on to, that are quiescent and spend longer in G0. So of course, the first thing we wondered was, could we use this biosensor to visualize cell cycle state in everyone's favorite model organism, C. elegans? And so we've made a variety of different constructs. These are all single copy constructs integrated into the genome at safe harbor sites at either chromosome 1 or chromosome 2 using CRISPR-Cas9 genome engineering. And so we've picked promoters that are expressed ubiquitously in all developmental stages. And so this is just showing that the biosensor is expressed in the embryo. And you see that you get nuclear exclusion in the embryo as cells are quickly cycling. And then it's also expressed in the germline. And if you have any questions about our biosensor design, please just put questions in the comments. I'm happy to answer them at the end, or if there's no time, I'm happy to reach out directly. We spent a lot of time sort of optimizing the biosensor and picking floors and testing different promoters. And so we learned a lot about ratiometric biosensor design in this way. For the rest of the time, I'll be showing you mostly data from the GFP version of our biosensor, as that was the brightest and easiest to image over long periods of time. And then we focused most of our time looking at some post-embryonic lineages, and that's because after birth, where you're born at about 558 cells, during the L2, many of these cells leave quiescence and then have to proliferate several times, these last lineages, to give rise to the adult structure. And so we focused on 3 lineages in particular: the sex myoblast, a pair of mesodermal cells that migrate down the length of the body and then divide 3 times to differentiate into vulval and uterine muscle; some lineages within the somatic gonad, the ventral uterine and somatic sheath lineages; and finally, a lineage that my lab spends a lot of time looking at, the vulval precursor cells, which divide several times to give rise to the adult vulva. And so here I'm just going to show you a movie of the biosensor where we follow one cell division in the worm. This is one of the vulval precursor cells shown here above the star, the yellow star. So it's going to divide. You'll see that right about here, divides, and you get 2 cells that are born with nuclear signal. And so you can see over time, you lose signal in the nucleus. You notice that the nucleolus never localizes, so you're really just looking sort of at a donut. The histone mask is shown here on the right. And so if you go ahead and plot those, you can see that the biosensor behaves as you'd expect, and that you see this increasing amount over S phase to G2, and then G1, and then it starts going up again. And so the question we can ask then is, so our biosensor behaves as we'd expect in a cell cycle dependent manner. And so the question we'd like to know is, can we use it to see the difference between a cell that's born and going to divide again versus a cell that's going to exit the cell cycle? And of course, C. elegans makes a particularly attractive model to test this because of the invariant lineage. And so here we're plotting several movies showing the proliferation of the sex myoblast cells. And so they're shown here on the top. So after they migrate down, they start cycling again. You can see that first cell division. And if we take the mean of the CDK activity, the ratio at the division, we get a number around 0.5. get this, a very similar number at their next cell division. These are both pre-terminal divisions, and it's this last division where we go from 4 to 8 cells on one side of the animal that these cells differentiate. And hopefully you can appreciate both from the micrograph where you see stronger nuclear localization, so that means lower CDK activity, and also the quantification, the mean value, that we get a statistically different value at terminal differentiation as these cells are born versus these cells pre-terminal differentiation. Okay, so that's the sex myoblast. We also have data from the uterus. It looks quite similar, but in the interest of time, I'm not going to show you because I'm going to spend the rest of the time talking about the division of the vulval precursor cells. The vulval lineage is quite nice because, like the sex myoblast, they also are born from 3 cells that will divide 3 times to give rise to 22 cells. And we'll talk about how we get to 22 cells in a second. But the first thing I'm going to show you is some stills from a movie looking at the first cell division. And so here you can see we start in G1, then we have metaphase, anaphase, and then as the 2 cells are born underneath the anchor cell, 2 cells right here, you see they have nuclear localization again in G1. And so if we plot that, you can see we get to a CDK2 increasing state, around very similar to the sex myoblast lineage, and then that starts going up again as these cells have to divide again. And so this next cell division is really interesting because as we go from 6 cells to 12 cells, what we see is that 2 cells exit the cell cycle right here, these 2 green cells, which I'll be talking about a lot. They're called the D cell or Vol D cell. And so the reason that you have 22 cells instead of 24 cells in the adult vulva is that these 2 D cells exit the cell cycle at this cell division where every other cell goes on to divide one more time. And so hopefully you can appreciate that the biosensor sees this cell. Here's the 2 D cells. We see strong nuclear localization in both D cells at their birth. And that if we plot this out, we can see here's the trace from all our D cell movies, and here's all the other cells that are going to go on to divide one more time, and then they divide and they exit with strong nuclear localization. And so very easily we can see a difference in the vulva between a cell that's exited the cell cycle and all its sister and cousin cells. And so from mammalian cell culture, Sabrina Spencer and others were able to show that a negative regulator of the cell cycle, so a cyclin-dependent kinase inhibitor, so in humans that's P21, and in worms there are 2 of them, CKI1 and CKI2, that these CKI levels actually increase in G2 and then peak in G0. And so if you look at a summary of her data from a recent PNAS paper from Sabrina's lab, you can see that in a cell that's going to divide again soon, you just get a little blip of CKI at G2, but then it remains relatively low. Whereas at the next division, when this cell is going to exit into a CDK2 low state, we see a increase in CKI or P21 activity that's followed by sustained burst of CKI as this cell is exited into a CDK low state. And so we wondered if similar dynamics were occurring in the worm. And so using CRISPR-Cas9, we inserted a GFP tag as a C terminus of one of the 2 CKIs in worms and CKI1. And then we took many static images. Unfortunately, the strain is too dim to time lapse due to photo bleaching, but we were able to collect many Z stacks and sort of compile a lineage and localization pattern from that. And so here I'll just take you through some of the micrographs and I'll show you the plot next. And so here we're just following the primary cells underneath the anchor cells, sort of marked here in this orange color. As you can see at G1, there's basically no expression of CKI. After, as these cells enter G2, we see some expression at G2 of CKI1. You can see the nice nuclear exclusion. These cells are in G2. And then these cells are, the next division is born at G1, and you can see, again, some expression of CKI1 at G1. And then these guys are going to divide one extra time. And so here I'm showing the innermost F cells at their prolonged G2, and you can see they're expressing CKI1 robustly. And then if we look later, here's that terminal division. So there'd be 4 cells on top and 4 cells on bottom. So I'm showing you the confocal slice. You can see 4 of those cells, those 4 primary cells, the F and E cells, and they're all in G0 and expressing CKI1 GFP. And so if we go ahead and plot using pseudotimes, just taking all of our time course data and allotting it over vulval development, we see a very similar pattern in that we see some increasing levels of CKI1 over the first cell cycle. This was the one whole cell division before terminal differentiation. And then at the next cell division, we see this peak at G2 before cells have divided into G0, and then we see this prolonged levels of CKI in G0. This was really exciting. So some of you may have noticed some interesting things in my image. So what that just shows, I'm going to go back and look at a different lineage. And so the first thing that you might wonder is, well, what does the D cell lineage look like? Because if you remember from a couple of slides ago, I told you that the D cell is born at this next division and terminally differentiates. And so here's the D cell mom. So this is the cell that's going to give rise to C and D, marked here in cyan. And so if we follow it at its birth, we see here that it's in G0. And you can see it has stronger nuclear localization than its sister cells, the primaries in G1, and it's expressing high levels of CKI. And so if we plot that out, we see here's the D cell, and so here's CKI activity as the D cell is born into G0, so it fits with our model that a terminally differentiated cell expresses high CKI levels. Now, if you're looking at this image, you may notice something interesting, something we noticed too, in that in this case, in these images, the sister C cell also looks like it's expressing strong nuclear localization, so low CDK activity of our biosensor and high levels of CKI1. And so, in fact, if we look later, what we find is that in this animal, for example, this C cell actually never divided. And so the first thing we started wondering is, did the insertion of our GFP tag create a gain of function phenotype? So as we know from now many labs doing these CRISPR insertions, it's just because you have a transgene that seems to rescue your mutation and behave normally the way you tag it doesn't mean that when you insert the tag into the native locus that it will be 100% wild type. And so what we found, and by looking through many examples, is that we see that in the case of the C lineage, that almost a completely penetrant phenotype where the C cell also exits into a CDK low state and does not divide, in fact, at 98% of the time. We saw this at a lower penetrance in some of the other vulval lineages. And so we're pretty sure what we're seeing is a gain of function phenotype where we're getting a failure to turn over sufficient amounts of the CKI1 protein from the insertion of the GFP tag. And so this is clearly interfering in a cell cycle dependent way. Now, this suggests that the CD lineage is particularly sensitive to cell cycle perturbation. And in some ways, this makes a lot of sense because as you remember, the CD cell mom gives rise to two cells, one that's going to go on to divide and one that's going to exit and stay out of the cell cycle forever. And so not surprisingly, both C and D cells are born with lower CDK activity than their sisters, but the C cell then goes right back into cell cycle. So looking through many movies, we noticed that in some rare cases, we saw D cells that were actually born with higher CDK activity than a normal G0 state. So on here on one side, you can see a C and D cell that are born with low CDK activity. And then on the right, you see that they have less nuclear signal. So this made us wonder, the first thing we wondered was this a byproduct of the insertion of our transgene? So many biosensors, of course, can have a phenotype. So the first thing we wanted to look at was, do we see this phenotype in all our strains of our biosensor? And so as I mentioned before, and I'm happy to answer questions before, we made a bunch of varieties of these biosensors under different promoters and different fluorophores. And here's the wild type expression pattern where we see strong nuclear localization in all of these strains of the D cell at its birth. But at anywhere from a 2 to 10% phenotype, when these animals were grown at 25 degrees C, we saw one cell out of two born with higher CDK activity. Now, we thought that this, again, still at this point, thought this might just be from the biosensor itself, but Jessica Feldman's lab, Maria Celia, postdoc in Jessica's lab, sent us an endogenously tagged DNA licensing factor, CDC1. So this is nuclear localized in cells that are at G1 or G0, and very nicely nuclear localized in the D cell at its birth. And in fact, in 1 out of 50 animals of CDC1 at 25 degrees, we saw the same thing, where one of the two D cells was born without nuclear CDC1, just like it was born into higher CDK activity state. And so what this tells us is that we see a potentially mitotic D cell in all of these strains, including strains that aren't expressing our biosensor. So it seems like it's likely not to be a biosensor phenotype. So I've had a lot of time during COVID-19 to sort of think this through. And so, of course, the worm community is amazing. At the New York area worm meeting when we spoke about this a couple months ago, Ivor Greenwald suggested that we reach out to Paul Sternberger and Marianne Felix. Of course, if you have any questions about vulval development, it's best to go right to the source. And so I sent an email a couple weeks ago to Paul and Marianne, and actually Paul answered in 2 hours, and he sent me to his original Cell paper from 1986 where he looked at the vulval lineage, and sure enough, I have it quoted here. He said, "We have observed some variability in the lineages generated by P5P and P7P." This is actually his thesis from the Horvitz lab, and that specifically at 25 degrees, he sees an extra cell division at some penetrance. And so this is in the literature from over 30 years ago, but certainly underappreciated. Now, those of you that know Marianne Felix will not be surprised that although she didn't reply in 2 hours, about 2 days later, I got a really lengthy email from her, including some unpublished data from her lab when Michaelis Barkoulis was a postdoc in her lab. He had was trying to look at stochastic cell divisions in a seam cell and was trying to compare them to stochastic divisions, extra divisions in other lineages, and he happened to look at the vulva. And so what he found at high penetrance is that if you take L2s and move them from 20 degrees to 28 degrees, so close to heat shock, he had about a 30% defect in extra D cells by lineaging these animals at the L4. And so this was really exciting because what it suggested is that our biosensor could be used to see a cryptic stochasticity. And I sort of put this slide in here because as a C. elegans biologist, I think lineage is really important to all of us. And it's sort of ironic that this is turned into a complete postdoc lineage because I'm sure many of you know Marianne Felix was a postdoc in Paul's lab, and Michaelis, who did that experiment, was a postdoc in Marianne's lab. And then one of Paul's other postdocs was Dave Sherwood, who was my postdoc mentor. And so then, like any good biologist, we can just map these onto our lineage to see that we both end up at the terminal D cell. So it's not surprising that we have both looked at this phenotype. And then, of course, we're just 2 generations removed through Paul from Sidney Brenner. Okay, so that was my C. elegans lineage digression, but back to our data. We saw cells born into a CDK higher state, so then we wanted to look through our movies. And so I'm going to play you a movie on the left here, and I have stills as well. And so here's the D cell. You can see that it starts losing CDK nuclear localization, so increasing, goes up. And then right here, we get that division. So here's our 2 daughter cells, and I'll break again. And then it's probably easier if I just play you through these stills. And so here's our D cell. It's already losing compared to its sister D cell, that nuclear localization. There, the chromatin's condensing there. And then in this case, we get a nice longitudinal division of 2 D cells born into G0. And so we found 10 movies where that happened, and we hand quantified that. And so if we plot that dividing D cell, we see that it looks an awful lot like a wild type sister C cell that normally goes on to divide. And so I told you in the beginning that Sabrina's lab was able to show that you get this bifurcation at mitotic exit in cell culture, where you can predict a cell's behavior in cell culture based by activity post-anaphase into 2 population cells that cycle again with CDK increasing state and a CDK low state where they are quiescent for some time. So we wanted to ask, could we do that with all of our traces? And so what we did is this represents all of the raw data from all the lineages we took in the SM cells, the uterus, and the vulva, and then we just overlaid on this data what the lineages are. So whether a cell in blue is exited in G0 versus a cell in yellow. And so this is really exciting. Right now, we're doing some modeling with Sabrina's lab to look at where in this curve post-anaphase we can make a call with predictability about what a cell is going to do. Right now, it looks like it may be as early in some lineages at 80% with 15 minutes post-anaphase, which is really exciting. And so I'm just going to end my talk with one example of how we're using this tool to answer some biology. And as you can imagine, you could come up with many ways to use this tool. And this is sort of why we got into this in the first place. And that is that I mentioned in the beginning that most of my lab studies the invasive capacity of the C. elegans anchor cell. And this, of course, is work I started in Dave Sherwood's lab as a postdoc. Of course, he started as a postdoc in Paul's lab. And so the anchor cell is a specialized uterine cell which invades into the vulval epithelium in a highly coordinated way based on the timing of the division of these underlying vulval cells that we just spent a lot of time looking at. And of course, it's been a really nice model for trying to understand how cells acquire the ability to become invasive. And when I was a postdoc in work I finished up at the beginning of my doctoral position at Stony Brook, is that we identified that a single transcription factor, when you take it out of the anchor cell, NHR67, a nuclear hormone receptor, causes the anchor cells to become mitotic. And that result of entering the cell cycle is inhibitory to invasion. And so that led us to a hypothesis that cell cycle arrest is important for invasive behavior. And so our model at the time was that the transcription factor's job was to positively regulate the activity of CDK1. This kept the anchor cell in G1, G0, and that downstream of this, a chromatin modifier, an HDAC, HGA1, was important for maintaining this differentiated state. And so in 2015, what we were missing was the ability to assess cell cycle state of these HDAC deficient anchor cells. And so we've gone back and done this now. And so this is work from a postdoc in the lab, Nick Palmisano. And so here showing a wild type anchor cell with an anchor cell marker, and then our cell cycle strain. Of course, wild type anchor cells are hanging out in a G1, G0 state, as you'd expect for a post-mitotic cell. NHR67 deficient anchor cells, so these represent these clusters of mitotic anchor cell tumors, so they're all over the cell cycle state. And then interestingly, we see both rare occasions where we get extra ACs and also many single AC blocks, that ACs that fail to invade, but these ACs, at least a portion of them, clearly are cycling. And so here we can use this tool to identify, even in cases where we just see a single blocked AC, that the AC is no longer in G0. Another graduate student in the lab, Jason Smith, has sort of built upon that, and we've done a large-scale chromatin modifier screen where we identified multiple members of the SWI/SNF complex that also seems to affect cell cycle state in the AC. And so I've just summarized some of his data here, where even in single blocked AC, so in this AC right here that's failed to invade, it actually is born with a non-G0 level of the biosensor. And so we get a large portion of single ACs that are cycling inappropriately. And this biosensor is perfect for seeing that sort of cryptic phenotype. And so that's forced us to sort of reaffirm a model that we published from a couple of years ago, that these chromatin modifiers likely function both at the level of cell cycle arrest and cell invasion. And lastly, I'll just say that I sort of mentioned in the beginning that I have sort of deep evolutionary developmental roots. And so we've been collaborating with lots of labs, putting this biosensor into as many organisms as we can, from xenopus to zebrafish with our next door lab neighbors, Ben Martin's lab, the slipper snail, Crepidula. A lot of these experiments were done in collaboration with the Woods Hole embryology course over many summers. It works in sea stars and nematocella and chick. Basically, every organism we've tried to use it in, it looks like it works. It's really exciting. And with that, of course, I'd like to thank all the lab members that past and present that have worked on this project, as well as all the current Matus lab members, and thanks for many of the funding sources. And with that, I'd be happy to take any questions.
All right, thank you very much for a fascinating talk. Let's see, we already have one question. Does the cell cycle regulator regulate cell invasion or mobility through activating mechanosensing signaling?
Does the cell cycle work? So if I understand the question correctly, you're wondering if the being in G0 regulates invasion. So what we found is that you need to be in G0 to regulate and to be invasive. So you actually don't need the transcription factor NHR67 here at all. But if we just overexpress CDK1 in an anchor cell that has the transcription factor depleted, so where we have mitotic ACs, just having CDK1 in the anchor cell alone, which puts the anchor cell back into G0, is enough to restore the invasive phenotype. So of course, we're very interested in trying to look at what's downstream of being in a G0 state that promotes invasion. And so doing RNA-seq, of course, would be sort of the tried and true in ATAC-seq to sort of get at the suite of targets. And those are things we're trying to work on now.
All right, thank you very much. I have a question. Like, is the extra division event, is it like a random effect in a population, or if you isolate the worm which have the extra division that it is transferred to the progeny?
Oh man, that is a great question that we have definitely not done. Yeah, so right, we're seeing this at about 2 to 10% clip when we push these animals under high stress, right? So under higher temperatures.
Mm-hmm.
So I think if we could get it to work, we found out about the 28 degree thing now under quarantine. So we want to obviously go back and push our worms to 28 degrees so we can get this up to hopefully 30%, because then it'd be a lot easier to rescue worms off of slides and check it out, because that'd be really interesting to know. Yeah.
Yeah. Okay. Thank you. Is there any other question for David? If you have some, and then we don't have the time to, you can ask them on our Slack, the elegans Slack channel. Other than that, thank you for a very great talk, you and Javiti. And